of two different plasmids and both replicate in the same cells is controlled by plasmid genes involved in regulating DNA replication. When a plasmid is transferred into a cell that already carries another plasmid, a common observation is that the second plasmid may not be maintained and it is lost during subsequent cell replication. The two plasmids are then said to be incompatible. A number of incompatibility groups have been recognized. The plasmid of one incompatibility group excluding each other but being able to coexist with plasmids from other groups. Plasmids of one incompatibility group share a common mechanism of regulating their replication and are thus related to one another. Therefore, although a bacteria cell may contain different kinds of plasmids, they are not closely related to because they must be compatible. Some plasmids called episomes have the ability to become integrated into the chromosome, and under such conditions their replication comes under control of the chromosome. This situation is remarkably like that of several viruses whose genome can become incorporated into the host genome. Plasmids can sometimes be eliminated from host cells by various treatments. This process, termed curing, apparently results from the inhibition of plasmid replication without parallel inhibition of chromosome replication, and as a result of cell division the plasmid is diluted out. Curing may occur spontaneously, but it is greatly increased by use of acridin dyes, which become inserted into DNA or other treatments that seem to interfere more with plasmid replication than with chromosome replication. Electroporation may also be used to cure a cell of plasmids. Many of these characteristics are exemplified in a very well characterized plasmid called F plasmid. The F plasmid is a circular DNA molecule of 99,159 base pairs. Cells containing it can be easily cured with acridin orange. Shows a genetic map of the F plasmid. One region of the plasmid contains genes involved in regulating DNA replication. It also contains a number of transposable elements involved in its ability to function as an isome. It has a large region, DNA, the TRA region, containing genes that permit it to be transferred from one cell to another. Cell-to-cell transfer plasmids. Because one of the defining characteristics of plasmid is lack of distinct extracellular form, one might imagine that the plasmids are confined almost exclusively to transferring only to daughter cells during cell division. Since some prokaryotic cells can take up free DNA from the environment, it is possible that the lysis of the host, however it may happen, may bring the plasmid in contact with the new host. However, this process occurs naturally in only a few bacteria species and is unlikely. To account for a much cell-to-cell plasmid transfer, the main mechanism of the cell-to-cell transfer is conjugation, and is a function encoded by some plasmids themselves. Conjugation is a replicative process, and both cells end with copies of the plasmid. Plasmids that govern their own transfer by cell-to-cell contact are called conjugative, but not all plasmids are conjugative. Transmissibility by conjugation is controlled by a set of genes with the plasmid called the TRA region. The TRA region contains genes encoding proteins that function in DNA transfer and replication, and others that function in mating pair formation. The presence of a TRA region in a plasmid can have another important consequence if the plasmids become integrated into the chromosome. In that case, the plasmids can mobilize the transfer of chromosomal DNA from one cell to another. Strains of bacteria that transfer large amounts of chromosomal DNA during conjugation are called high frequency of recombination. The use of conjugation to transfer host genes is discussed further in the next section. Some conjugated plasmids from Pseudomonas have a broad host range, that is, they are transferable to a wide variety of other gram-negative bacteria. Some conjugation plasmids can transfer genetic information between distantly related organisms. Conjugated plasmids have been shown to transfer between gram-negative and gram-positive bacteria. Between bacteria and plant cells, and between bacteria and fungi, even if the plasmid cannot replicate in the new host, the transfer of the DNA itself could have important evolutionary consequences, as well as been involved in the pathogenic process, if it can recombine into the genome of the new host. Clearly, all plasmids must carry genes. Types of plasmids and their biological significance. Clearly, all plasmids must carry genes that ensure their own replication. However, for many plasmids we know very little about what other genes they carry. These are called cryptic plasmids, and they were discovered by physical means, such as examining a cell extract using gel electrophoresis. As we have seen, some plasmids also carry genes necessary for conjugation, and they can sometimes be detected biologically either by the transfer functions themselves or by sensitivities to the certain viruses. Although plasmids do not carry genes that are essential to the host under all conditions, the presence of plasmids in cells can have a profound influence on the cell's phenotype. In some cases, plasmids encode properties that we think of as fundamental to the bacterium in question. For example, the ability of rhizobium to interact with the plants. Some plasmids and Pseudomonas have been shown to transfer the genetic information for biochemical pathways that degrade unusual organic compounds such as camp or octane and naphthalene. Plasmids can carry a very wide variety of genes. Indeed, the only limitation is that the genes they carry not interfere either with their own replication or with the survival of the host. Because plasmids can carry large and may carry many different genes, it is not always a simple matter to classify a plasmid into a simple phenotype category. As we shall discuss, a single plasmid may confer many different phenotypes on its host cell. A few of the phenotypes conferred on the prokaryote of plasmids are given. It is likely that virtually all prokaryotic groups possess plasmids. In the remainder of this section, we will discuss a few of the many phenotype plasmids may confer only cells. Resistant plasmids. Among the most widespread and the well-studied groups of plasmids are the resistant plasmids. Resistant plasmids which confer resistance to antibiotics are various other inhibitors of growth. Our plasmids were first discovered in Japan, and strains of enteric bacteria that had acquired resistance to a number of antibiotic multiple resistance have since been found throughout the world. The emergence of bacteria resistance to several antibiotics is of considerable medical significance and was correlated with the increasing use of antibiotics for the treatment of infectious disease. Soon after these resistant strains were isolated, it was shown that they could transfer resistance to sensitive strains via cell-to-cell contact. The infectious nature of the conjugated R plasmids permits rapid spread of the characteristic through the populations. A variety of antibiotic-resistant genes can be carried by F plasmids in general. These genes encode proteins that either inactivate the antibiotic or affect its uptake into a cell. Plasmids R100, for example, is 94.3 kilobase pair plasmid. that carries resistant genes for sulfonamides, streptomycin, and spectinomycin, fusidic acid, chloromiphenyl, tetracycline. R100 also carries several genes conferring resistance to mercury. R100 can transfer itself between enteric bacteria and the general Acinetobacter, Klebsiella, Proteus, Salmonella, and Shigella, but does not transfer to the non-enteric bacterium Pseudomonas, or plasmid with genes for resistance to most antibiotics are known. Many drug-resistant elements are plasmids, such as those on R100, are transposable elements. Section 10.11 and this plus the fact many of these plasmids are conjugated make these plasmids a threat to traditional antibiotic therapies. Toxins and other virulent characteristics. We will discuss the physiological and genetic characteristics of microorganisms that enable them to colonize, host, and set up infections, which can lead to harm. In the present context, we merely note the two major characteristics involved in virulence: the ability of microorganisms to attach to and colonize specific sites, and the host. The formation of substances, toxin enzymes, and other molecules that cause damage. The host. It has now been well established that in several pathogenic bacteria, each of the virulent characteristics is carried out plasmids. For example, electropathogenic strains of Acinetobacter coli are characterized by the ability to colonize the small intestine and to produce a toxin that causes symptoms of diarrhea. Colonization requires the presence of cell surface protein called the colonization factor, antigen, CFA, encoded by the plasmid, which confers on the cells the ability to attach the epithelial cells of the intestine. At least two toxins of enteropathogenic E. coli are known to be coded by plasmid: the hemolysin, which lyses red blood cells, and the ectrotoxin, which induces extensive secretion of water and salts into the bowel. It is the enterotoxin that is responsible for the induction of diarrhea, as well we discussed. Pathogenesis in terms the causative agent of plaque requires the expression and secretion outer proteins, antigen and the V antigen. These proteins have functions that allow the bacterium to subvert host defense functions. The genes that encode that regulate these proteins are carried by a virulence plasmid. Some virulent factors are known to be encoded on the plasmids, while others are known to be encoded by other types of mobile genetic elements, transposons and bacteriophages. Several examples are now known where genes producing a particular type of infection are present on different genetic elements within the same cell. For instance, the genes encoding the virulent determinants of the